Welcome. In this video, I'd like to talk about chromosomes, specifically the relationship between lots of terms related to chromosomes: DNA molecule, unreplicated chromosome, replicated chromosome, sister chromatids, homologous chromosomes, non-sister chromatids, and non-homologous chromosomes. I'd also like to talk about the way that chromosome structure changes throughout the cell cycle. So when you think of chromosomes, you might think of something that looks like this, but really chromosomes only exist like that at certain points in the cell cycle. And most of the time, chromosomes look more like this in your cells, but that's not very easy to draw or think about, so we'll use some models to depict the way that chromosomes look throughout the cell cycle. So I'd first like to start with just the definition of a DNA molecule. So when I say DNA molecule, what I'm talking about is a very long double strand of DNA. So this would be one DNA molecule, a double helix. That's somewhat hard to draw, so sometimes you will see this depicted as a ladder. Same thing, one DNA molecule. For this video, I don't need to really think about the two different strands of DNA. So when I draw one DNA molecule, I'm just going to draw one line with a centromere. So all of these are the same thing. So one DNA molecule, just keep in mind, for this video is one line, but it is a double helix. And at most points of the cell cycle, really this DNA molecule is uncondensed and floating around in the nucleus so that genes can be read from this DNA molecule. So one DNA molecule at certain points in the cell cycle is called a chromosome, and specifically it's called an unreplicated chromosome. This unreplicated chromosome is not just DNA, it's wrapped around proteins to help it from getting tangled, but we're not going to depict those in this video. So in G1 phase of the cell cycle, each chromosome is unreplicated and consists of one DNA molecule. These DNA molecules are very, very, very long, millions of base pairs in our cells, but we are going to depict them like this. So this is one unreplicated chromosome. Now during S phase of the cell cycle, DNA replication happens, and specifically semi-conservative DNA replication. And this is this replication bubble that you know and love, where the two individual strands of DNA separate from each other, and each is used as template to make a new DNA strand. So what you end up with after semi-conservative DNA replication is two identical DNA molecules. And I'm just going to use one line to depict the DNA molecules. So these are two identical DNA molecules connected to each other along their whole length, but we'll just draw the centromere here. So the relationship between these two identical DNA molecules these are called sister chromatids. Now we know that there are genes along this whole length of this chromosome, and because they're identical, let's just put the A gene up here in this specific spot. If there is a big A allele on this sister, because they're identical, of course this sister should also have a big A allele. So remember that when we draw something like this, what that really consists of is two identical DNA molecules that are a result of semi-conservative replication. Now this is also called one chromosome. I know that's a little confusing, but that's just the terminology. This is called one replicated chromosome. So we've got one chromosome before DNA replication and one replicated chromosome after DNA replication. And now we know what sister chromatids are. So I think you know that in your cells you have chromosomes inherited from your mom and chromosomes inherited from your dad. The relationship between these are called homologous chromosomes. So for example, let's say that this is chromosome 1 that you've inherited from your mom. We call this the maternal copy. And I'm going to use a different color pen to look at the relationship between your maternal copy of this chromosome and your paternal copy. And I'm going to draw the paternal copy already replicated. So the paternal copy meaning it's inherited from your dad. The relationship between these, they're actually very, very similar to each other. They have the same genes in the same positions, but they may have different alleles or different versions of those genes. And when I mean different alleles, these genes are 99% the same, but the 1 or so percent or less percent difference results in different expression or different shaped proteins. So the way I'm going to depict that here, I'm going to put the A gene in the same spot on these 2 chromosomes, but I'm going to do a different allele. I'm going to do a little a allele. In the relationship between these 2 chromosomes, they are called homologous. Homologous meaning the same. They are very, very similar to each other. One is inherited from your mom, one is inherited from your dad. And notice that these are separate chromosomes. They are always separate in every cell of your body. The DNA that you inherit from your mom and the DNA you inherit from your dad remain as separate DNA molecules in all of your body cells. So one other term I'd like to introduce right now is non-sister chromatids. So non-sister chromatids are chromatids from a homologous pair. So the relationship between this DNA molecule here and this DNA molecule here. These are called non-sister chromatids. From the same homolog, but they aren't sisters. Or from the same homologous pair, I'm sorry, but not sisters. So the last term I'd like to talk about in this video is non-homologous chromosomes. And what these are chromosomes that are not similar to each other at all. So if this is chromosome 1 that you've inherited from your mom, and this is chromosome 1 that you've inherited from your dad, I think you know that you've got 23 different chromosomes. So let's look at chromosome 2, for example. So chromosome 2 that you've inherited from your mother, let's just look at the replicated form, will have totally different genes on it than chromosome 1. So I'm just going to put the B gene on it. These chromosomes are, in addition to having different genes, are different sizes. So they're not similar at all. And let's just draw the homolog that you've inherited from your dad for chromosome 2. There we go. So the relationship between chromosome 1 and chromosome 2 is that these are non-homologous. So if we summarize and look at the way that chromosomes change throughout the cell cycle, we can do that here. In G1 phase, each chromosome consists of one long DNA molecule. And I'm going to use pink to look at the 2 chromosomes in our example that were inherited from mom. And there's the corresponding chromosome inherited from dad. I'm going to go ahead and put centromeres on here too. So right now we've got 4 chromosomes and a total of 4 DNA molecules. So during S phase, these chromosomes replicate semi-conservatively, so that in G2 phase of the cell cycle, what you end up with is that each chromosome consists of 2 identical sister chromatids held together. And remember that the alleles on sister chromatids should be the same because these DNA molecules are identical. So here in G2 phase of the cell cycle, we have a total of 8 DNA molecules, but still 4 chromosomes, 4 replicated chromosomes. And as the cell prepares for cell division, nothing will happen to the DNA except that it will condense. So we're drawing these chromosomes like this, but really they are uncondensed and your cell would look more like this. This would be a tangled mess during cell division if it wasn't condensed before the cell divides. So all that happens as the cell prepares for cell division is that these chromosomes condense. Nothing different about the DNA, they're just condensed and ready for cell division. So your next video will be about what happens during specifically the cell division meiosis to form gametes, sperm and egg.